Welcome to this lecture on genetics, and we will cover in detail the process of reproducing life and genetic variation. So, let's get started. In my last lecture, I ended with a brief introduction to DNA. Well, what exactly is DNA? What is it made of, and how can a small molecule like DNA code for all of the traits in a living organism? I will address these and other questions in this lecture. Ultimately, what we are doing in this lecture is understanding how the genetic code, or DNA, results in variation, because it is this variation that natural selection can act on and lead to evolutionary changes. We will start by looking at the fundamental unit of all life on Earth, the cell. Inside each cell, the DNA code is structured into packages known as chromosomes. We will see how the DNA molecule can copy itself so that each cell in an organism's body contains the same DNA information. We will then look at how DNA codes for proteins, which all living organisms are made of. Finally, we will look at a concrete example of how DNA affects our lives by examining human blood types. Though we have to dive into the microscopic world, don't lose sight of the big picture. DNA is a code for making proteins, and we are made of proteins. If the DNA changes slightly through mutation, which we discussed in the last chapter, the protein changes and thus the organism can change too. All living organisms are made of cells. The cell is the basic unit of life. There are many organisms that are made of just one cell, and many, including you, that are made of trillions of cells, and that's amazing. All of life can be divided into two big categories, depending on the kind of cell they have. The first kind of organisms are called prokaryotes. Prokaryotes are single-celled bacteria without nuclei or any special structures we call organelles. They often have structures shown here in this image, like a cell wall, an outer membrane, a cytoplasm within which the DNA resides, and they often have locomotor functions like a flagellum. Shown below is a microscopic image of a prokaryotic cell that we all have heard of, E. coli, which lives in the guts of many mammals, including humans. Though prokaryotic cells live within us and have been instrumental factors in driving human evolution, we will turn now to the cells we are made of, eukaryotic cells. All animals, plants, fungi, and many single-celled organisms called protists are made of eukaryotic cells. These cells have a nucleus that contains DNA and often have membrane-bound parts of the cell called organelles. These include chloroplast, such as those found in plants, and mitochondria, which help produce the molecular energy that powers cellular processes. Notice in this image that the eukaryotic cell is a bit more complicated than the prokaryotic cell. The microscopic image here is of kidney cells, which clearly have a DNA nucleus that contains a membrane that keeps the components of the cell contained, and a fluid in the cell called a cytoplasm. There are two types of eukaryotic cells in all animals and plants: somatic cells and gametes. Let's start with the somatic cells. Somatic cells, also called body cells, are found all over the body. Shown in the image above are the somatic cells found in the heart, brain, nerves, blood, bone, and skin. Somatic cells all contain a complete copy of the organism's DNA. For example, in humans, somatic cells have all of the DNA packed in 46 chromosomes. Somatic cells also replicate through a process called mitosis, which we will learn about in just a moment. The other kind of cell in the body is a gamete. Gametes are the sex cells. At the right are gametes, sperm cells and egg cells. The large round cell at the bottom right is an egg, or ovum, and the plural of these are ova. The small wiggly structures surrounding the egg are sperm. These gametes contain only half of the organism's DNA, which are 23 chromosomes in humans, and replicate through a process known as meiosis. It makes sense that each of these gametes contain only 23 chromosomes. When they fuse together during conception, the chromosomes can be added together to form 46, the number found in somatic cells. Since we just mentioned chromosomes, and we can see this in the image at the bottom left of the slide, it is worth examining chromosome numbers in a bit more detail. Humans have 46 chromosomes in our somatic cells, 23 from our mother and 23 from our father, for a grand total of 46. But the number 46 is not at all special. Other apes, like chimpanzees, have 48 chromosomes. Some primates have fewer chromosomes, like the colobus monkey, which has 44. Some organisms that we would consider to be less complex than humans have fewer chromosomes, like the housefly with 12, or the salamander of 24. But plenty of organisms have more than we have, like the potato with 48, the camel with 70, or algae, which has 148 chromosomes. Classifying organisms by the number of chromosomes they have would be like organizing books in the library based on the number of pages they have, or by the color of their jacket covers. It wouldn't make sense. What matters are not the number of chromosomes an organism has, but a similarity in DNA that is packaged in the chromosomes. For instance, humans and chimpanzees, though having a different number of chromosomes, share about 98% of their DNA code. This is remarkable and in some way indicates how important a 2% difference can be. You most likely all heard of DNA and have probably heard that it is the blueprint or recipe or code for life. But how does this work? It helps first to understand the structure of DNA and to understand how it is packaged in your cells. It is estimated that there are the equivalent of 6 feet of DNA in every cell in your body. Think about this, 6 feet in every cell of your body. If cells are microscopic, how can this be? As shown in this image, the DNA molecule is wound up into compact structures that we have already encountered, chromosomes. Sections of that DNA code for specific proteins your body makes and needs. These sections of DNA are called genes. All of the genes put together in all of the chromosomes are called the genome. The DNA that is in the nucleus of our cells is called homoplasmic, meaning that it is more or less the same in every cell in our body. But the nucleus is not the only part of a cell that contains DNA. At the bottom left is an image of an organelle called the mitochondrion, or plural is mitochondria, which contains its own DNA. Mitochondrial DNA is much, much smaller. It contains only 37 genes. Unlike DNA, mitochondrial DNA can differ from cell to cell, making it heteroplasmic. And these genes are inherited only from your mother, meaning that they can be used to trace one's maternal lineage, called a matriline. In fact, scientists have used mitochondrial DNA types, called haplogroups, to figure out when and how humans migrated from northern Asia into the Americas. Scientists have been able to extract many small pieces of ancient mitochondrial DNA from the skeletons of the earliest Americans, amplifying it using a technique called polymerase chain reaction, or PCR, and study the genes of humans who died thousands of years ago. Combined with information from living Native Americans, it is now hypothesized that the colonization of the Americas happened at least 15,000 years ago and consisted of humans with 4 different mitochondrial DNA sequences. These include the haplogroups A, B, C, and D. DNA is just not the tool for understanding migration patterns. It is the code for making you. It is time to discuss what DNA actually is. DNA is a molecule, in fact, a very simple one. DNA is made of 3 things: a type of sugar, a phosphate group, and a nucleotide base pair. The sugar and phosphate form the backbone of the long DNA molecule and do not vary along in the chain. What varies along the chain are the nucleotide base pairs. These bases can be of one of 4 types: adenine, or A, thymine, just T, guanine, which is G, and cytosine, which is C. You can think of DNA as a ladder, with the sugar and phosphates forming the uprights and the bases forming the rungs. These rungs are made of two base pairs that cling together using hydrogen bonds. Critical to understanding DNA is the fact that the base pairs do not randomly cling to each other. Instead, A only clings to T and C only clings to G. These are called complementary bases. What this means is that if you know of one side of the DNA chain, you know the other. If a DNA sequence is CAAT, the other side must be GTTTA. Though any two humans may have over 99% of their DNA base pairs order identical, there are of course differences. Differences in these single nucleotide regions are called SNPs, or pronounced SNPs, in short for single nucleotide polymorphisms. Because these vary from individual to individual, there are some of the areas of the genome that can be used to solve crimes using DNA. One of the most astounding accomplishments of modern science has been the decoding of the entire human genome. The precise sequence of nucleotide base pairs, the A's, G's, C's, and T's, that make up a typical human have been mapped. This was not an easy job, since there are 3 billion nucleotide base pairs in the entire human genome. Surprisingly, scientists found that humans do not have the 100,000 plus genes expected, but rather only 25,000 to 30,000. For instance, on the right is a map of chromosome 3, with many different genes of different nucleotide base pair lengths identified. With this new information, scientists have begun to mine the human genome to figure out precisely which genes do what, and which genes are implicated in some human diseases. One of these diseases is osteoporosis, which results in the thinning of bone with age and an increased likelihood of fracture. By studying osteoporosis in humans and in a primate model, such as baboons, scientists have found a gene on chromosome 11 that increases the risk of bone loss. But how do genes affect bone density? Let's return to our model of DNA to see how it actually works. The very structure of DNA explains how it is replicated so easy and so accurately. When a cell is going to divide, it copies its entire genome. Remember these nucleotide base pairs? Well, there are 3 billion of them to copy each time a cell divides. And cells divide all the time. It happens when you went from a single fertilized zygote to 2 cells, then 4, 8, 16, until you were several trillion cells worth of a newborn baby. It continues to happen as old cells divide to form replacement cells. The DNA replicates faithfully every time. DNA replicates so easily and so accurately because of those A's, G's, C's, and T's we discussed a moment ago. A double-strand DNA molecule is unwound by enzymes, and the hydrogen bonds connecting the complementary base pairs are broken. What results are 2 template strands that have so-called sticky ends, free-floating nucleotide base pairs in the nucleus of the cell, which are acquired through the foods we eat, which have their own DNA too, and these bind to the template strands following the rule of these base pairs. A goes with T and C goes with G. In this case, CTAT is separated from GATA. These sticky ends become templates to form 2 new strands that are identical to one another. Again, it is the very structure of DNA that explains how copies of it can be made. We often say that the replication process produces identical copies of DNA, but that is not entirely true. Copy mistakes can occasionally occur, and this is yet another source of genetic variation. Before we get into the nuts and bolts of mitosis, the name for somatic cell division, let's consider those chromosomes one last time. In order to make sure that each copy of the cell sequence of DNA gets into each cell, the chromosomes must pair up and replicate. 23 of these chromosomes were inherited from the mother and 23 from the father, and each chromosome number, 1 to 23, is different in its length and in the genes it contains. These chromosomes pair up into matching or homologous pairings in the somatic cells. Though these chromosome pairs, shown in these images, may look identical, they may well contain different versions of a gene, known as alleles, since one chromosome was inherited from the mother and the other from the father. 22 of the 23 homologous pairs of chromosomes are referred to as autosomes. The 23rd pair determines the sex of the individual and is appropriately named the sex chromosomes X and Y. Females have 2 X chromosomes, one inherited from their mother and one from their father. Males have an X and a Y, the X from their mother and the Y from their father. Because females have 2 X chromosomes, they only contribute an X in the egg cell they produce. Because the male has both an X and a Y, there is a 50-50 chance that a sperm will contain an X or a Y. It is therefore true that the male determines the sex of the child by contributing either an X, and therefore producing a female, or a Y, and therefore producing a male, though this is not a conscious decision that sperm cells make. One way to visualize the homologous chromosome is the product that is called a karyotype, which are illustrated here on the slide. Notice the 22 homologous pairs of autosomes numbered according to their size, and last, the sex chromosomes. Based on what you see here, what karyotype is from the female, which is from the male. If you look closely here, you'll see that the top has 2 X's and that's a female, and the bottom illustration is a male because of the X and the Y chromosomes. He started as a single fertilized egg called a zygote. It had 46 chromosomes. Cells with this full set of chromosomes are called diploid. As we'll soon see, cells with half the number of chromosomes, or 23, are called haploid. These are the egg and sperm cells, and there is a very good reason that they have half the number of chromosomes, which we'll encounter in just a moment. But back to this zygote. During embryological development, this little zygote divided to form 2, 4, 8, 16, 32, 64, and eventually trillions of cells. These cells soon form tissues and organs in a process known as embryological development. If the DNA divided as the cells do, your chromosomes would go from 46 to 23 to 11.5 to 5.75. Of course, this did not happen. Instead, every cell contains 46 chromosomes. This occurs because, as I have already discussed, DNA can replicate itself and does so before each cell division, so that the cell goes from 46 chromosomes to 92 before dividing into 2 cells, each with 46 chromosomes. This process by which this occurs is called mitosis. As shown in this figure, mitosis starts with a single diploid cell that has 23 pairs of homologous chromosomes, or 46 chromosomes. These chromosomes duplicate by unwinding their DNA and attaching free nucleotide bases to the template strands in the manner already discussed. After chromosome duplication, the cells technically have 92 chromosomes, which all line up in the middle of the cells, so that one full set is on one side and another full set is on the other side of the cell midline. These cells pull apart into 2 daughter cells, each with identical DNA. The microscopic image is of skin cells divided into 2 daughter cells. Each of these cells has 23 pairs of homologous chromosomes, or for a total of 46. If an egg cell had 46 chromosomes and a sperm had 46 chromosomes, the resulting zygote would have 92 chromosomes. This simply would not work. So gametes would have to divide up the DNA a bit differently than somatic cells do. Instead of having a full copy of an organism's DNA, gametes are haploid, meaning that they contain only one chromosome for each pair. The one chromosome can be the one inherited from the mother or the one inherited from the father. With 23 chromosomes, the number of different combinations is exceptionally high, meaning that each egg and each sperm cell contains a unique combination of genes from the organism's mother and father, introducing yet more potential for variation. The process by which this occurs is called meiosis. Meiosis starts the same way as mitosis. The DNA replicates and the homologous chromosomes pair up. The cells then divide into two identical daughter cells, just as happens in mitosis. However, unlike mitosis, the cells then divide again, resulting in 4 daughter cells with 23 chromosomes each, but no pairs. Right before that final cell division, the homologous chromosomes can recombine their chromosomes in a process called crossing over. A chunk of chromosome 2 from the mother's line can switch with a chunk of chromosome 2 from the father's line. So the 23 chromosomes in the gametes are not only a random assortment of chromosomes from the mother and from the father. Within each chromosome, there will be a combination of genes from the individual's mother and father, genes that are close together on a chromosome, therefore tend to move together and cluster together. These clusters of genes are called haplotypes, which can be used to access the history of genetic lineages, which I discussed earlier in the context of the peopling of the Americas. If chunks of DNA are exchanged on non-homologous chromosomes, called translocations, diseases such as leukemia can result. If the chromosomes fail to divide, the resulting gametes can have too few or too many chromosomes. Too few can result in a mesomony, and too many in a trisomony. Down syndrome is an example of a trisomony in which there are 3 rather than 2 copies of chromosome 21. We've obviously been learning about DNA and chromosomes. In the last lecture, we learned about Mendel and his genetic experiments on pea plants. What do the two have to do with one another? This is a very important slide that demonstrates how these concepts are linked. Here's another Punnett square in which 2 pea plants with identical genotypes and phenotypes are crossed. Each plant has yellow seeds in a green pod, and each plant is heterozygous. Remember that this means that both plants have each allele for pod color and seed color, but that the dominant allele is expressed. It would be expected that the offspring of breeding identical plants like this would be identical to the parents, but genetics does not work that way. During meiosis, the top pea plant will produce a gamete, an egg or sperm, with either big G or little g combined with either big Y or little y. Each has an equal chance of being produced, resulting in 4 possible combinations of genes in the gametes: GY, big G, big Y, big G, little g, big Y, and little gy. The same applies for the plant on the left of the Punnett square. Now when these gametes are combined together in all possible ways to produce zygotes, the resulting baby plants will have the genotypes shown on the Punnett square and a 9:3 to 3:1 ratio of phenotypes. 9 will have green pods and yellow seeds like the parents, 3 will have green pods and green seeds, 3 will have yellow pods and yellow seeds, and 1 will have yellow pods and green seeds, which is the exact opposite of what the parents had. Notice that having one particular color of pea pod had nothing to do with the color of the seed. This is known as a law of independent assortment. Notice also that the rules of genetics and the processes of meiosis produce plentiful variation, the raw material for natural selection to act upon. But these are pea plants. What about humans? Here's an example that is more applicable to you and me. Suppose the gene for hair color is on the small chromosome and the gene for eye color is on the large chromosome. The blue allele on the small chromosome represents blonde hair and the large chromosome represents blue eyes. The red allele on the small chromosome represents brown hair and on the large chromosome, brown eyes. Both parents are heterozygous, and we'll say for the sake of argument that brown eyes and hair are dominant, meaning that both parents have brown hair and brown eyes. The parent on the left, which is female, produces 4 eggs through meiosis. Because of the law of independent assortment, the alleles for hair and eye color are independent from one another, producing 2 eggs that pass on the alleles for brown hair and brown eyes, and 2 eggs with the alleles for blonde hair and blue eyes. This is just as likely what happens with the male. He produces 4 sperm cells, 2 with brown eyes and blonde hair, and 2 with blue eyes and brown hair. Choose 1 egg and 1 sperm and combine them. What do you get? Now choose another. Notice that there will be a mixture of these features. Some of the offspring will have blonde hair and brown eyes, some will have brown hair and blue eyes. Keep in mind that some of these combinations were not present in the parents. Some of you may be saying that hair color and eye color are not independent, but they do seem to be present together. Brown with brown, blonde with blue. You're right. One of the reasons for this is that some of the genes that code for these traits are, in fact, on the same chromosome. The bottom image shows genes close to one another on the same chromosome will not follow the law of independent assortment and will instead be linked to one another. This is called linkage. How does DNA cause a seed to be yellow or hair to be brown? When we talk about DNA as a code, what do we exactly mean? Besides replicating itself, the other critically important thing DNA does is to code for proteins. Proteins are what bodies are made of. There are 7 types of proteins described here. Some, called enzymes, help with chemical reactions such as the protein lactase, which helps break down the lactose, sugar in milk. Others are structural proteins, like the keratin that makes up our hair and nails, or the collagen that helps make up bone. These are shown in this image to the right, with keratin to the top and collagen at the bottom. There are gas transport proteins like hemoglobin, which transport oxygen throughout the body. Antibodies, which help fight diseases, are proteins. Hormones like insulin, which helps regulate the metabolism of sugar and fats in the body, are proteins. Muscles are composed of the mechanical proteins actin and myosin. Finally, protein can be the nutrient form, like ovalbumin, which is found in egg whites. Proteins are critical for the normal functioning of an organism. How does DNA code for these proteins? First, it is important to recognize that proteins are made up of amino acids. There are 20 different kinds of amino acid. Humans can manufacture 12 of these. The other 8 have to be eaten and are therefore called essential amino acids. These 20 different amino acids can combine into chains of various lengths and different properties. These properties are what makes a protein like keratin different from a protein like hemoglobin. The 20 different kinds of amino acids that make up proteins are listed in this table. How does your body make these proteins? It uses DNA as a recipe. Alanine is added if the DNA code is CGA, CGG, CGT, or CGC. Tryptophan is added if the DNA sequence is ACC. Look at the chart. Which sequences would cause lysine to be added? What about glutamine? Notice that the amino acid being added to the protein chain is coded by DNA in groups of 3 nucleotide base pairs. These are called codons. Let's delve into more detail and look at how this actually happens. Protein synthesis, or the process by which a DNA code is turned into a chain of amino acids, occurs in cells. First, the DNA code is read by enzymes, producing a molecule called messenger RNA. This process, in which messenger RNA is created from a DNA code, is called transcription. The messenger RNA then leaves the nucleus of the cell and enters a cytoplasm. It binds to ribosomes, which are organelles that facilitate the translation of messenger RNA into a chain of amino acids, which ultimately form a protein. Just like during DNA replication, the DNA is unwound or unzipped by enzymes. However, unlike replication, only one of the strands of DNA is used during transcription. Also, unlike replication, only a specific section of the DNA is unwound. This region is called a gene. The unwound DNA strand serves as a template for making a single-strand molecule of messenger RNA. RNA is very similar to DNA, but instead of using A, G, C, and T as base pairs, RNA uses A, G, C, and U. U stands for uracil, and it binds to adenine, A, just like thymine, T, does in DNA. As is shown here, if a gene has a sequence of TACTC, the messenger RNA molecule will be AUGAG, and so on. Once the gene is fully transcribed, the messenger RNA molecule leaves the nucleus and finds ribosomes in the cytoplasm of the cell. Once messenger RNA binds to a ribosome, translation of the code into amino acids can begin. As we have already seen, this process occurs in threes. Three nucleotides, called a codon, are read by the ribosome by matching a complementary anticodon to the codon. For instance, if the messenger RNA codon is AUG, then the anticodon has to be UAC, since those are the three nucleotides that are complementary to the codon. It is important to note that these anticodons are attached to a very specific amino acid, methionine in this particular case, in a structure called transfer RNA, or tRNA. The next three nucleotides in the codon are AGU, which match with the anticodon UCA, which is attached to the amino acid serine. This goes on in groups of three until the last codon, UAG, which is a stop sequence. The amino acid chain is then released into the cytoplasm. The amino acid chain folds into a three-dimensional structure or bonds with other 3D proteins, which gives these proteins their specific properties. What we have described here happens in only a small percentage of the human genome. In fact, only about 5% of the total genome is composed of structural genes that code for proteins. Much of the rest of the genome was once regarded as junk DNA. However, we now know that this junk DNA is not entirely junk. Much of it is composed of regulatory genes, which turns genes on and off. Regulatory genes can be thought of as on-off switches, or perhaps more accurately as dimmer switches. Regulatory genes determine if a gene is on or off and can regulate the amount of protein produced and when. For instance, if the genes controlling connective tissue growth are left on a bit longer during development, what can result are longer, thinner fingers, among other traits, as shown in this image. This is characteristic of a disease called Marfan syndrome, something that some scholars think Abraham Lincoln may have had, shown on the right. Regulatory genes have also allowed us to understand major evolutionary events. For instance, paleontological evidence demonstrates that modern birds evolved from a group of feathered dinosaurs. But anyone who has visited a science museum knows that dinosaurs had teeth. Birds do not. Where did their teeth go? Scientists have recently discovered that birds still have the structural genes to make teeth, but the regulatory genes controlling these structural genes have been turned off. A similar thing has happened with human body hair. Humans have less body hair than other primates. We still have the genes for full body hair coverage, but these genes have been down-regulated. Similarly, all baby mammals have the ability to digest milk. This is because they produce the enzyme lactase, which breaks down lactose. However, these genes are turned off in most adult mammals. However, some humans have lactase persistence, meaning that the genes are not turned off and they can continue to digest milk as adults. Those who retain the typical mammalian condition of losing lactase production into adulthood are said to be lactose intolerant. Notice that natural selection can act on the products of structural genes, and it can also operate on the products of variation in regulatory genes. In fact, research on human and chimpanzee genomes have discovered that while structural genes are very similar, there are important differences in those regulatory genes. One of the best examples of regulatory genes are those of the homeotic or Hox family of genes. These are master switches that determine the general form of an animal's body. Notice that whether you are human, a mouse, or a fruit fly, heads are where heads should be, bodies are where bodies should be, and limbs are where limbs should be. Why is this? Researchers have discovered that a group of genes called the HOX genes, or HOX genes, regulate the position of the major body parts during embryological development. What was amazing to researchers was that the very same genes regulate this process of body formation in organisms as different as flies, mice, and humans. Small changes in how long these genes are switched on or where they are expressed can result in differences in overall body form. For instance, the genes of the neck region are positioned differently in birds and snakes, giving birds long necks and snakes short necks, but long bodies. The HOX genes that determine forelimb and finger length are switched on for a longer period of development in bats compared to other animals. Again, selection can favor the product of variation in regulatory genes as effectively as structural genes. Let's look at one more Punnett square to consider how variations in specific genes can result in even more possible combinations of traits. As I have already discussed, the mother and father contribute equally to the genetic makeup of the offspring. This is known as a law of segregation. Consider the example in which a pure red sweet pea is crossed with a pure white sweet pea. The offspring in the first generation will all be heterozygous, meaning that they will inherit the red allele from one parent and the white allele from the other. If the resulting flowers are all red, then the red allele is said to be dominant over the white allele. But what if the flowers are all pink? This can happen. It means that these two alleles are both expressed, neither is dominant over the other, and they are said to be co-dominant. If these flowers are crossed, the offspring will be a combination of pure red, which is a genotype big R, pure white, genotype of little r, and pink, which is heterozygous genotype big R, little r, or shown here as hybrid red. Let's apply these principles to humans. Human blood type is a good example of a trait with multiple alleles. A person can be blood type O, A, B, or AB. Because there can be more than one kind of blood type, this is referred to as a polymorphic trait. What do these blood types mean in the terms of genetics? Every person inherits two forms or alleles of the blood type gene, one from the mom and one from the dad. These can be allele A, B, or O. There is no such thing as an AB allele, yet they can have AB blood type. How? The A allele codes for the protein that we call A. The B allele codes for a protein that we call B. If someone has the A allele on one chromosome and the B allele on the other, they are said to have blood type AB. This is because these alleles are co-dominant and both blood proteins are produced. So, do people with the O allele make an O protein? No. In fact, they do not make a protein at all. This is why the O blood type is referred to as the universal donor. Since there are no proteins on the surface of the cells, the recipient of this blood type will not attack those cells with antibodies. Somebody with blood type AB does not make antibodies against either A or B, and therefore can receive blood from any blood type. However, someone with blood type A will make antibodies against B and cannot receive that blood type without fatal complications. Likewise, someone with blood type O makes antibodies against all other blood types and cannot receive any other blood type except O. Let's look at this again in the context of genetics and try to answer the following questions. Can parents with blood type A and blood type B have a baby with blood type O? The answer is yes. Draw a Punnett square to try to figure this out. Can either of the parents be homozygous, or must they both be heterozygous? Try this one. Can a man with a blood type AB be the father of a child with blood type O? Why or why not? By this time, you are probably realizing that genetics is a complicated business. In fact, it is much more complicated than I've described in this lecture. Many traits are polygenic, meaning that multiple genes are responsible for the phenotype observed. For instance, a person's height or skin coloration can be influenced by hundreds of different genes. In addition, these and many other traits can be highly influenced by the environment. For instance, height can be strongly affected by nutrition. Remember that natural selection can only work on traits that are passed on from one generation to the next. So, quantifying the role that genetics has in shaping a particular phenotype can be quite important in determining the role of natural selection in shaping it. Complicating matters even further is the reality that the same gene can influence many different phenotypes. The sickle cell gene, for instance, influences both the individual's ability to combat malaria and the ability to transport oxygen throughout the body. It turns out that most traits are polygenic and most genes are pleiotropic, modeled here at the bottom right, making genetics a fascinating if quite complicated science. Furthermore, recent research has revealed that the nature, genetics, or nurture environment question is fallacious. All traits are a product of both genes and the environment. Additionally, there's evidence that environmental factors can actually alter the regulation of DNA itself, a phenomenon known as epigenetics. Don't lose sight of the big picture, though. Our journey into the world of genetics should have revealed to you two of the key tenets of Darwinian natural selection: the heritability of certain features and the source of variation. In the next chapter, we'll look at how gene frequencies can change over time.